j-ISBA. Okay. Called Advances from Bayesian Statisticians. I'm just gonna read the introduction. So I'm sharing. So thanks everybody for joining this fourth annual of the International Day of Women in Statistics and Data Science Conference with this year's theme, Thriving in Your Environment. We are recording the event and the videos will be available on the Caucus for Women in Statistics YouTube channel. We kindly ask you to post your questions to the speakers in the Q&A chat. And without further ado, I'm delighted to host this session, Advances from Bayesian Statisticians, S308, and to introduce the first speaker, who is Beatrice Franzolini. Beatrice is a tenure track assistant professor in Milan Bicocca. Beatrice, I think you can share your screen. And you can start. Thank you very much.
Thank you, Francesca, and thanks to the organizer for this great event. I hope you can hear me well and see my screen. I will try to tell you a little bit about some recent work I've done with some collaborators about dynamic Bayesian mixture models. I would like to start quite informally though, telling you a little bit about the idea that is behind basically random partition modeling in general. So I guess whatever application, statistical application you may have in mind, it should be very easy to think about the observational units in your data set. Here, these 10 circles represent ideally 10 observational units. In practice, hopefully we have far more than 10. But the idea of random partition modeling basically consists with the idea of adding flexibility to basically any parametric model we may have in mind for the observations collected over these observational units. And the way to do that is to assume that there is some latent clustering configuration of the observational units. And then such that within each cluster of these observational units, we can use some cluster-specific parametric model. Of course, if we do this and we are not too careful, what may happen is that we may introduce too much flexibility. And of course, we don't want, for instance, to degenerate on some model where each subject is in his own cluster because we could not learn anything from a model of that kind. So in general, there may be some problem of overfitting when you try to apply this strategy. And one way to control for overfitting is, for instance, to take a Bayesian approach. That is what I'm about to do. Because there the prior acts as a penalty against overfitting. But so what happens at that point is that basically any latent objects and any parameter will have its own distribution, its own prior, and so the clustering configuration, the partition will be random because it will have its own distribution. Just to become a little bit more effective with an example, the classical example of this modeling strategy are Bayesian mixture models, for instance, normal mixture models. And what happens in that case that we model our observations as some random sample, and our likelihood is a mixture model, for instance, of normals with unitary variances. And the idea here is that if we take a Bayesian approach to this, there is an equivalent way to write down this model, which is indeed the recipe that I was trying to present informally. It is to define a random partition, this row, which is indeed the clustering of the subjects that is going to have its own distribution. And then conditionally on this random partition, what is left to do is to define some parametric model for each cluster in the random partition. So the idea of this talk is that I will try to tell you a little bit how to extend this recipe of having one random partition and then parametric models, one for each cluster in the partition, to when our data are repeated measures data. So for instance, panel data in a longitudinal study, or also crossover studies, or even multi-view data. In all these cases, what happens is that we're still going to have our observational units, but now instead of having just one observation for each of the subjects, we are going to have a bunch of them. So let me use as a working example, in particular, panel data. So we have our subjects here in the different rows of our data matrix. And then when I move across columns, what happens is that I'm moving through time. And so for each subject, I'm actually collecting observations across time. So I think that before going to how we can do this with a dynamic Bayesian mixture model, there is an obvious question to answer. That is, can we not just use what is already out there and was already studied and well established, like standard Bayesian mixture models? So one way to do that, let's say, to do that, what we can do is to consider each row in this data matrix, so the observation for one subject, as an observation, a vector observation. And then a very general way to write down a Bayesian mixture model is as this expression here in the slide, where, for instance, we have to choose some kernel that would be multivariate because our observations are vectors, for instance, a multivariate normal. So where is the random partition here? So the idea is that we have to pick some prior for the parameter of this model, which is this random probability p tilde, which is the mixing measure in the mixture. And if we choose this random probability to be almost surely discrete, so to be just a discrete distribution, what happens is that the likelihood simplify to the expression we had already in the first slide. So it's just a discrete mixture. And basically, if we have this discrete mixture, we can rewrite the model as a random partition model. So there is a random partition here. So what is going on? What is this random partition doing here? The random partition is clustering the different rows of our data matrix, which can be fine sometimes, but other times we may have many problems with this. And the idea is that there may be problems in interpretation of results if we are interested in the clustering. There may be problems in the modeling the dynamics of this data and also in the estimation per se. It may be tough to come up with a point estimate of this partition of these clustering configurations. Here are some references with some example of these problems. But I will not go too much into detail about them. If you're interested, those are the references. So we need something else. We need some random partition model that is not really clustering the rows of a data matrix if we are working with panel data. And so there is one proposal. There was one proposal in the literature when I started working on these topics. And basically the idea is that since we know that if we pick a mixture model and we choose a mixing measure that is almost surely discrete, what we are doing is to induce a partition. What we can do if we want even more flexibility and not to cluster just the rows is to use one random probability measure for each time point, because in that way we will come up with the one partition for each time point. And there are a lot of models, especially in Bayesian non-parametrics, to do so. But when they are used for panel data, they have some drawbacks. And I think a way to see the drawbacks are these two results. So the first one concerns the clustering mechanism that is underlying these models. So whatever fancy can be this prior over p tilde, any model that can be represented in this way will learn the clustering, will learn the partitions, assuming that the probability that subject I and j are clustered together at time t is going to be independent on whether they were clustered together at another time point compared if they were not, basically independent. So this is an equality, which I don't think is necessarily what we want to do if we really want to model some dynamics in the partition. Even probably more convincing results is what happens if we look at the predictive distribution in time. So models of this kind, what happens here is that the prediction that we're going to make for a certain subject I, is going to be the same that we do for subject j, even though these two subjects may follow completely different trajectories in our data set. So these two drawbacks, we were not the first to notice. There are already a different level of generalities that were already noticed in the literature. So now I will tell you what is our proposal, our solution to this, and how we propose to do dynamic mixture models. So these smiley faces are again our observational units, what we want to cluster and partition. And we are at time point 1, and we're going to use some random partition model because for just one time point, those models are well established. But then the idea that we came up with, and we call the conditional partial exchangeability, or CPE, is the following. When we move to the second time point, the idea here is that we are not going to use just one random probability measure to induce a partition, but we're going to use many of them, one for each of the previous clusters. And I'm trying to skip technicalities, but basically the idea is that this will work basically for a generalization of Cauchy-Schwarz inequality. And the idea is that since those red subjects are associated with the common random probability measure that is different from the others, they are going to be clustered together more often since they were already clustered together. So this is a slide with a full model for panel data with many time points constructed based on this idea of CPE. The only thing that I will just make 2 comments about this. The first one is that here what happens at time point t, we can see that the random probability measure controlling the mixture model depends not only on the time point t, as in the negative example I showed before, but also on m. And what is m is an indicator that tells me where this specific subject was allocated at the previous time point. And basically this trick is what allows us to have in terms of clustering and random partition, a learning mechanism that I think is the desirable one to capture the dynamic of a random partition. And now subject I and j will be clustered together at time point t with higher probability in the case they were already clustered together compared if they were not. So I will just show you a simulation study to show you that this makes sense. And here what is going on is that we are simulating data for 200 items, 200 subjects, observational units, and just 10 time points. There are 2 underlying clusters, 2 underlying groups. One are simulated from a normal distribution centered in 0, and the other from a normal distribution centered in 4. These 2 normal distributions have unitary variances, so they are very well separated. If you plot the data, you basically can see the 2 clusters, so it's very easy to estimate the 2 clusters. And then from one time point to another, we just move 10 observations from one cluster to the other. Here are the results. I'm going to comment to the first panel here, the RAND index. The RAND index tells us how good we are recovering the true partition, and when it's equal to 1, we have perfect recovery, and the recovery deteriorates as the RAND index goes down to 0. So the one in bolts are the models developed under this CPE approach that we are proposing. In the first column inside there is k-means. And as we can see, k-means here is estimated independent at each time point. It does very well. The 2 clusters are well separated. It's not surprising. But here the point, and why I'm showing this very trivial simulation study, is that those 2 models that are based on our approach, even in this case, we have some gain, and we can improve the recovery of the truth. And the idea is that we can borrow information locally and allocate subjects based on where they were also at other time points. Since I'm running out of time, that would be it. I had just some real data, but I guess I made my main point. Those are some references, and thank you for listening.
Thank you very much, Beatrice. Very interesting. So I don't see questions in the Q&A. Just remind everybody attending that you can write your questions there. If there are no other questions, I can... there, Daria raised it. No, she didn't. Very quickly, can you also think of models that model completely random measures or like random partitions through time with Markov chains, sort of like Markov processes?
Yeah, so underlying this one, there is a little bit a Markov chain across the different partitions. So yeah, I think
okay,
yes.
so it's already there. Okay,
Yeah.
Thank you very much.
Thank you.
Thank you, Beatrice, again. If anybody has questions, put them in the Q&A. So next speaker is Daria Bystrova. She's a data scientist specializing in causal inference at the Pierre-Louis Institute of Epidemiology and Public Health. Daria, do you want to share your screen?
Yep. I think it should be good. You can see my screen, right?
Coming up, yes.
Okay,
yes, we can.
great.
Thank you.
Thank you, Francesca, and thank you for invitation. It's great to be here. So I will present today the causal discovery from ecological time series. It's a joint work with Charles Sad, Sorenzi and Wilfred Thuyer. I'm sorry. So the idea of this work comes from the ecology. In ecology, we often want to understand the relation between different components of ecological system, for example, between birds and other insects and environmental characteristics. And the useful way to represent this relationship is using the causal graphs. So we want to understand the causal graph of this relationship. And under the causal graph in this presentation, I will assume directly the cyclic graph, where the vertices represent the variables of interest and the edges here represent the causal relationship. So for example, here y1 could be a species or environment, and y1, if we have a directed edge between y1 and y2, it means that y1 is causing y2. And in order to relate the causal graph to the data, we need to assume causal Markov condition, which states that every variable is independent of its non-descendants given its parents. And I will use this sign for statistical independence. So their causal discovery aimed to discover the causal graph or parts of it from observational data. There exist many families of causal discovery methods, and we focus today on a constraint-based family, which used conditional dependence tests for discovering causal graphs from data. And these families need several assumptions. The first assumption is faithfulness, which states that all conditional dependencies in the distribution are entailed by causal Markov condition. And the second assumption is causal sufficiency, which states that there is no hidden confounding. The problem with using conditional dependence tests for discovering causal graphs is that actually several causal graphs have the same conditional dependence pattern. So if you look at the simple graphs on the left, where x goes in y or y goes in x, they actually have the same conditional dependence pattern. They are marginally dependent. So if we look at the set of graphs below, where we have 3 nodes and in each of the structures, nodes x and y are dependent. However, if we condition on the middle node z, they become conditionally independent. And finally, the third structure that is important for this presentation is unshielded collider. Here, if in this structure x and y are independent, however, if we condition on middle node z, they become dependent. So it was proven that 2 causal graphs are Markov equivalent if and only if they have the same skeleton, meaning that the same set of unrelated edges and the same unshielded colliders. And all equivalent graphs can be represented by a completed partially oriented acyclic graph, which is called CPDAG. And as you can see from the name, its graph contains directed and undirected edges. So now I'll present one of the most known algorithms of this family, PC algorithm. So it works in 2 steps. And first, we construct the skeleton. I remind it's a set of undirected edges. So we start with a fully connected graph, and then we firstly remove all the edges that using marginal dependencies, and then we remove the consecutive edges using conditional dependence tests by progressively increasing the size of a conditional dependence set, of a separation set. And the second step of PC algorithm is orientation. After we remove all the edges, we progress to orientation. And firstly, we orient all unshielded colliders. And after we orient all unshielded colliders, we progressively orient using the contradiction to having all unshielded colliders or acyclicity. So it was proven that PC algorithm is able to find correct CPDAG of the true DAG under the causal sufficiency and faithfulness. There also exists extension of the PC algorithm that doesn't require causal sufficiency, which is called FCI. And although I mentioned it in the presentation, I wouldn't really describe it. It is a more complex algorithm, and it's have a more complex output than CPDAG. So coming back to our setting, in this work, we wanted to understand what will happen if we apply constraint-based causal discovery for ecological data. And we consider the following setting. So let's assume we want to sample different locations and we want to measure species abundances, such as counts, for example, and some environmental characteristics. So if we have multiple locations, these green points represent each location, let's say in France. So we sample each location, and in reality, in each location, we have a dynamic ecological system, let's say for 3 species. The problem that due to economical constraints, we actually often sample only once and stay for simplicity to the last point. So if we want to represent the ecological relationship, causal relationship in the ecological system, we will use full-time causal graph if we have a temporal observations. However, the problem that in our case, we do not have temporal observations, we only have 1 point. So our sample consists of in location, one sample in each location. And when we work with a full-time causal graph, in general, we would like to work with a summary graph, which is a causal abstraction, and summarize the causal relationship in the graph. It is way easier to work with. And in general, if we have a temporal data, we are able to recover directly summary graph from the observational data under specific assumptions. The problem that in our case, we do not have the full time observations, we only have the last time step. And the question that we ask, are we able to recover summary graph from this type of data? And the obvious problem that you can see that here, the hidden past of dynamical system work at the hidden confounding. So we'll answer this question under a specific, under additional assumptions. So we assume that there's a consistency throughout time interaction strength, meaning that causal relation doesn't change through time. And all variables are self-causal, meaning that for each variable, its past affects its next state. So what can PC and FCI find from this type of data? And as intuition probably can tell you that it is not able to infer much. However, we find that some interesting structure can be still found. So to define the structure, we first define super unshielded collider. So if you remember, I was already introduced you unshielded colliders. So here, super unshielded collider is the following structure, which is based on unshielded collider. However, Y2 and Y3 are marginally independent. In unshielded collider, it is possible that there exists a variable that are connected to both of them. However, they should not have edge between them. So after defining super unshielded collider, I can define cluster super unshielded collider. So what we assume by this? So if we have the following structure where in now each node represents the cluster. So how do we understand this cluster? It means that in each cluster, the variable, at least one variable in cluster Y2, Y4, Y1, at least one variable in cluster Y1, Y6, or cluster Y3, Y5, together, taking this one variable from each cluster, they will have to form super unshielded collider. And we found that given point-in-time observational distribution, the PC algorithm and FCI algorithm are only able to find all different components of the graph and all cluster super unshielded colliders. So we actually find that we can simplify a lot PC algorithm because steps 1.3 and 2.2 of PC algorithm is not needed because we actually only can focus on the marginal dependencies and only orient unshielded colliders. So in moving forward to our application, in real data, we often have different data types. And in ecology, we often have counts for species abundances and continuous variables for environment, such as temperature. So we chose to deal with mixed data using Gaussian-Koppel model. So here we have our observed vector y, and we model the dependence using the Latin variable z that have a multivariate normal distribution with correlation matrix C. And we relate our observed variables to Latin variables through pseudo-inverse of a combinatorial distribution function. So we use Bayesian-Koppel in we use Gaussian-Koppel in Bayesian framework, and so we want to use Gibbs sampling based on the extended rank likelihood introduced by Peter Hoff with the following prior for correlation matrix. So we use projected inverse Fisher transform for correlation matrix. So we simply sample covariance matrix from the inverse Fisher distribution, and then we transform it to the correlation matrix for the following expression. So we use this model for our application. And also we confirm our theoretical results and on statistical and ecological process-based simulation. I decided that it will be more interesting to see the real application. And as a real application, we have a French breeding bird survey. So where 30 species were sampled across 1,300 locations, and we also collected 8 environmental measurements such as temperature or precipitation. And the question we want to know for this data is what can we infer if we're only using one time stamp in this case. So the problem with this when we use our application that we don't have a ground truth. So luckily for this data set, we also observe 2 points in time. And so what we're going to do, we apply PC on 1 point in time data that should return us, in theory, cluster superunsheltered colliders. And we also use a 2 point in time data that under satisfied assumption, if we apply PC algorithm on this data, we should return the correct CPDAG. So the idea is to compare these two results. So how do we do that? So for each setting, for 1 point in time data and 2 point in time, we sample correlation matrix from the posterior distribution. Then for each of the sample correlation matrix, we apply the PC algorithm on it. So we will obtain total M graphs. Then we have to compute the final graph by keeping the edges that appear with a probability higher than the threshold beta. In our case, we took 0.8. And then we have to deduce a summary graph from the final graph. And so what we get, so we are comparing the orientations that are not related to the cluster superunsheltered colliders and orientation that belong to cluster superunsheltered collider. So we found that if we take orientations that are not in the cluster, we only can recover 8% of them. And if we take orientation related to cluster superunsheltered collider, we can recover 75%. So what our result seems to confirm is that cluster superunsheltered colliders are the most stable structure. So I move to conclusion. The causal discovery in general is a complex task and has been criticized a lot for unsatisfactory results in practical applications. So what we show is how constraint-based causal discovery can fail in inferring causal graphs from the specific 1 point in time data, which is quite often happens in ecology. And our point is to say that in this case, if you have this data, it doesn't make sense to compare the result of the causal discovery algorithm on this data with the true causal graph. Instead, it makes sense to compare to understand how well it is able to recover cluster superunsheltered colliders. And moreover, these structures can be found using a way simpler algorithm, which is based on marginal dependencies. And we also show that on real data, this type of structure seems to be easier to recover. Thank you for your attention. I'm ready to take any question.
Thank you, Daria. Maybe I can. I'm very ignorant about these type of models. So, what are the are there like, let's say, competitors? So you say that it doesn't make sense, you know, to look at like time steps separately, but are there models that try to model this time dependence already?
No, of course, it makes sense to tell them. The problem that we don't have these observations. The problem that we often, in reality, we often sample multiple locations once. So we were just wanting to understand what will happen in this case. Of course, if we observe the full time series, there's multiple methods that can discover the causal graph from the under additional assumptions, but still it can recover the causal graph from this data.
Okay, so yours is the first, like figuring out how to do it without observing the rest of the time series.
We wanted to understand what happens when you apply these methods on the data that we often have. And actually, we found that yes, if you applied the algorithm, you wouldn't infer much. And we actually understand that you can just use marginal dependencies, and you can still recover something useful. And we're working on how to make it useful, for example, for causal effect estimation. But we find that even the simple model can give you something. And actually, in fact, it's even better to use the simple model just using marginal dependencies because it is way more robust than more complex algorithm, but the result will be exactly the same. The result, the simple model will be more robust because it doesn't have to do multiple conditional dependence tests, and they can make extra mistakes.
Okay, thank you very much. Any other question? If not, we can move to Yunshan. So Daria, you can stop your screen sharing. So Yunshan Duan is our last speaker. We thank her because she's doing her postdoc in Johns Hopkins University, which means that it's very early in the morning for her. So you can start sharing your screen.
I hope you can see my screen.
Yeah, we can see it. Thank you very much. You can start.
Thank you, Francesca, and thanks to the organizer for this fantastic event. I'm very happy to be here, and today I'm going to share with you this project on spatially aligned random partition models with applications on spatial transcriptomics data. And this work is being done during my PhD at UT Austin and the supervision of my supervisor, Peter, and with my collaborators at MD Anderson Cancer Center. I'm going to start with an introduction of the motivating application of our proposed method. So in genomic data, in investigating the tumor microenvironment, we often observe this phenomenon in this following figure. For example, we have blue and green cells being immune cells and the red dots as tumor cells. And we often see groups of immune cells being recruited by a specific region of the red tumor cells. And we want to understand the mechanism behind this phenomenon. So we would like to aim to identify the subtypes within the tumor immune stroma cells that are potentially co-localized in this tissue slice to understand the interactions across these cell subtypes. The way of solving this problem, this is our proposed pipeline. We start with the observed data, single cell data and the spatial transcriptomics data. And with the help of spatial cell mapping method as a pre-processing, we get spatially enriched single cells, where for each single cell, we have the gene expressions and the inferred spatial locations on the tissue slice. With the help of biomarkers, we can split these cells into three types, tumor, stroma, and immune. And we aim to identify the spatially aligned clusters within these three types of cells, and we proceed with model-based clustering. For simplification of this explanation, we focus on two types of cells, non-immune and immune cells in this presentation, and the extension of the model into three types of cells are straightforward and could be found in our paper. To identify the spatially aligned clusters for these two types of experimental units, first we introduce some notations j equal to 1 and 2 to denote the non-immune cell and immune cells. For the observed data yji, we assumed being sampled from a mixture of kernel F with respect to discrete random matter Gj for type J cell. And to complete the construction of the model, we need to specify a prior probability model for those two related random matters G1 and G2. To construct dependent prior probability models on G1 and G2, there has been a lot of literature on dependent Bayesian nonparametric mixture models. This is a very limited list of some selected references, but we would like to argue that our problem has a very different setting and focus as the previous methods. To better understand the model structure that are desired for our inference problem, I generated this toy data set for illustration. Here, the blue dots are the non-immune cells, and the green dots are the immune cells. And we plot these cells with respect to the spatial coordinates on the tissue slice on the left-hand side. And on the right-hand side, we have the gene expression UMAP scores. We can see that we expect to observe and we would like to understand how co-localized of these clusters of the immune and non-immune cells. We often see non-immune cell regions clusters recruiting different clusters of immune cells. However, no matter how spatially co-localized it could be on the tissue slice, in nature, the gene expression profiles of the non-immune and immune cells could be totally different. In order to achieve this, we propose a spatially aligned random partition model. And on this slide, we can see the model construction, and first focusing on yji, denoting only the spatial coordinates. for J equal to 1 and 2 as non-immune and immune cells. We assume the spatial coordinates YJIs are being sampled from a mixture of kernel F with respect to discrete random matter GJ for type J. We construct the priors for G1 and G2 based on P-UR processes. For G1, we assume a vanilla P-UR process with parameters A1, sigma 1, and the base matter G1 star. Whereas for G2, we assume the prior based on P-UR process conditional on theta 1s, which are the cluster-specific parameters of the non-immune cells. That is saying that conditional on theta 1s, G2 is assumed to be sampled from a P-UR process with these parameters and a base matter G2 star having a specific structure here in equation 1. We assume G2 star is proportional to a mixture of G2 tilde, which is a vanilla non-atomic prior, and a mixture of kernel K centered at one of the locations at theta 1i, which is one of the locations of the non-immune cell clusters. This is saying that the atoms generated from G2 could either be as new locations from a non-atomic prior G tilde, or being sampled from a kernel centered at one of the non-immune cell cluster locations. So the second term in equation 1 allows for the spatial alignment of the immune cell clusters around the tumor clusters. This is the key part of our construction. But here on the slides, we only consider the spatial coordinates. And we want to extend our model to the full observed features, assuming Y including both the spatial coordinates and the gene expression PCA scores of the single cells. Because we only want to introduce the alignment in the subvector of the features only on the spatial coordinates, we assume the sampling model YJI being independent kernel Fs and Fg for the spatial coordinates and the gene expressions respectively. Here we introduce a triple of parameters theta being in superscript S, theta 1 and theta 2, where theta S indexes the sampling model for the spatial variable, and theta J indexes the model for the gene expression for cell type J. We can see that only theta S and theta J matching the subscripts are used in the sampling model, and the other parameter introduced in the triple of parameters is only for the introduction of the model construction, so that the theta lies in the same space for both J equal to 1 and 2 different type of cells that could be sampled using the discrete random matters on the previous page here. So this completes the model construction of the spatially aligned random partition model. And I'm going to skip the simulation studies and the model properties and jump into the real data analysis. Here's the data set we have on colorectal cancer. And I plotted out the real data after pre-processing. So these are already the spatially enriched single cells. Each of the dots denotes a single cell, colored by the annotated cell type being tumor, stroma, or immune. On the left-hand side, we have the single cell plotted with respect to the spatial coordinates. So these are how these single cells are actually located on the tissue slice. And on the right-hand side, we have the gene expression UMAP scores for these single cells. Applying our proposed method on the colorectal cancer data, we obtain these clusters for tumor, immune, and stroma cells. And looking at this plot with respect to the gene expression UMAP scores, we can see each of the clusters are homogeneous in gene expressions, which characterize the cell subtypes in nature. And to further understand how these cell subtypes are colocalized with each other on the tumor slice, we plotted out a selected set of clusters we identified for better visualization and plot these cells on the top of the histology image of the tissue slice. So we can see in these regions, we have those stroma and immune cells being located around the green and blue tumor regions. To further understand the mechanism behind, we also did some post-processing on the cell subtypes we identified, looking at the differential expressed genes of these immune cells that are specifically located around some tumor regions. And we find some interesting biomarkers that's validated in other clinical studies, which could be informative to understand the mechanism of tumor progression. In summary, we propose this spatially aligned random partition model that could be used on single cell and spatial transcriptomic data. The spatial alignment is formalized as the dependence in the spatial coordinate subvector to reflect the causal nature of the underlying biology of having tumor cells recruiting immune cells. And there's no notion of dependence in the subvector of gene expressions. And these are some selected references. Thank you for listening.
Thank you very much, Yunshan. Are there any questions? In the meantime, I can ask you something rather technical, which is you're using a Pitman-Yor process prior. Did you make some like sensitivity studies to figure out like what's the impact of that prior? Do you compare it to others? Does it like make some biological sense to use it, or
Yes.
it's just convenient?
Yeah, thank you. That's a great question. So we actually did carefully consider the prior choice, and we specifically used Pitman-Yor process with the Bocconi parameter sigmas as a negative value, which means this is actually a finite mixture in practice when on the real data. And one of the biggest reasons we use that is in practice, there's only a limited set of subtypes we can identify in the immune cells, or saying that we can, in the statistic model, we can go into a very high resolution of identifying the subtypes. However, it's hard to explain it or understand it biologically. So we limited the number of clusters we would like to identify by putting this hyperparameter setting. And we also did some simulation sensitivity analysis, choosing different parameters for the priors, and the performance is quite robust.
Okay, thank you very much. So thanks again to our three speakers, Beatrice, Daria, and Yunshan, for such insightful talks. Thanks to the organizer, which is IDWSDS, of course, and thanks to the junior section of the International Society of Bayesian Analysis, which is the sponsor of this session. Thanks to the joiners, to the attendees, and I guess I just invite everybody to stay over for the next session and have a happy International Day of Women in Statistics and Data Science.
Thank you, Francesca.
Thank you.
Thank you.